In this experiment here, we're looking at the coronary artery. The coronary artery is the main artery that feeds the heart. That's a very important blood vessel because it brings blood to the heart muscle, to the pump. And we have a ring with endothelium, actually 2 rings with endothelium, and 1 ring without endothelium. And then we contract it, we give it a little bit gasoline so that we can look at relaxation. I said that, I think, last time. And then we give increasing concentrations of noradrenaline. Note that this experiment is performed in the presence of a chemical that blocks beta-adrenergic receptors. So, if you remember vaguely what we said before, noradrenaline can act on alpha adrenoceptors and beta adrenoceptors. Here, this experiment, it cannot act on beta adrenoceptor because we have an antagonist, an inhibitor present. So we're looking at an alpha adrenergic response, if any. Okay? And you see that in the ring with endothelium, noradrenaline causes relaxation, while in the ring without endothelium it does not. And if we repeat the experiment in a ring with endothelium, but with a blocker of alpha-2 adrenoceptors, no response. Again, you don't have to remember all the details of this experiment. What I want you to remember is that if noradrenaline or adrenaline are in the bloodstream, if the adrenal glands augment their secretion, as we discussed in the lectures on homeostasis, the first cell that the adrenaline will meet are the endothelial cells. And in the coronary arteries, this will lead to vasodilatation. And again, this is very logical because when will we have more noradrenaline and adrenaline? In conditions of stress, when our cardiac function has to augment, when our cardiac output has to augment. You know, when we exercise, for example, we want more blood to the body, so the pump has to work harder. And at that time, you want the coronary arteries to dilate so that the perfusion of the heart is optimal. You understand that? So that's a very logical thing. Likewise, you remember we talked about antidiuretic hormone or vasopressin. And actually the next slide you've seen already. In this slide we compared the effects of increasing concentration of vasopressin in a cerebral artery, an artery of the brain, and a peripheral artery of the leg. And we did that in rings without and in rings with endothelium. And in the artery of the leg, there is no difference between the two. And vasopressin only causes a very strong contraction, illustrating that in peripheral arteries, vasopressin merits its name. Vasopressin means vasopressor, vasoconstrictor, increasing blood pressure. Okay? But if you look at the cerebral artery, we don't see a constriction. We only see a dilatation if the endothelium is present. Now from this slide, you can retain very important messages. One is that you cannot take one artery and extrapolate to the other. Each artery in our body has its specific way of doing things. In certain arteries, the endothelium is very important, in others it is not. Now this slide here illustrates something that I already said before when we talked about homeostasis, is actually a very logical thing again. Because vasopressin will be released in large amounts when the blood pressure acutely decreases. Remember, I told you that, for example, if we start losing blood, our life is in danger. At that time, we have two key missions. Try to keep the black box perfused and the pump working, okay? And this helps, this mechanism helps, because in the periphery we shut off blood for a moment, doesn't matter. We can live 20 minutes, half an hour without blood to our leg, it will survive. The coronary arteries remain open so that the pump can do whatever it does, and we have an opening of the big tubes to the black box, to the computer, so that whatever blood volume is still available, it can be redirected to the brain. Do you understand how beautiful this is? This logic, and all of that is decided in nature by these, specific behavior of the different blood vessels. So, those were two examples of a action on acute action of hormones on endothelial cells. As I told you, there also are chronic actions, and the best example came from this experiment where we took female rabbits, took out their ovaries, and then in one group did nothing else. In the other group, we reintroduced physiological levels of estrogens. And at this moment, at this time, all I want you to remember right now, I'll come back to that slide later, but you can see that progressively that the female hormone, the major female hormone, is potentiating endothelium-dependent relaxation. Now, this was the first time this was described, and in the meantime this has been reproduced by us and by many other people, and we know that chronic impregnation with estrogens will upregulate the presence and the activity of endothelial NO synthase and thus augment the production of NO in the arteries. Of course, we'll come back to that later. Good. So, hormones, acute and chronic. Remember what alkaloids were? Histamine and bradykinin, yes. Okay, good. Well, histamine, here you see relaxations to increasing concentrations of physiological concentrations of histamine, a very profound relaxation, but only in preparations with endothelium, not in preparations without. So the vasodilator effect of histamine that I mentioned already 2 or 3 times is basically due to its ability to cause the release of nitric oxide and EDHF in smaller arteries. At the level of the veins, I told you it does not do that, it causes constriction. Like we saw in the previous slide, if there is no endothelium, it's a constrictor. At the veins, the endothelial cells do not respond to this. So histamine will open up the arterioles and will constrict the veins. You remember this leads to an increase in hydrostatic pressure in the capillaries. At the same time, histamine makes more holes or increases the permeability, and we have the fluid that goes to the tissue. Remember, when you scratch yourself or the mosquito bites you, it becomes red because of this, and it swells because of that. Okay? If you don't understand that now after me explaining it 3 times, I don't know what I have to do. Okay. Good. So that's one example. Another very important example is bradykinin. In this experiment here, we looked at the cerebral arteries with and without endothelium. We pre-contracted it with, actually it was not phenylephrine in the basal artery, it doesn't matter. We pre-contracted it and then added increasing concentrations of bradykinin. And as you can see, there is a very clear-cut relaxation in rings with endothelium, but none in the absence of endothelium. Bradykinin is a very powerful endothelium-dependent vasodilator in most arteries, not in all species, but in most arteries it is. And in certain arteries, like the one that we were looking at, if we give an inhibitor of nitric oxide, oxyhemoglobin is something that scavenges nitric oxide. It picks it up, so the nitric oxide cannot get to the smooth muscle. This would be arteries with endothelium exposed to bradykinin under controlled conditions and in the presence of the blocker of nitric oxide. There is nothing left. In this artery, bradykinin causes endothelium-dependent relaxation only because it causes the release of nitric oxide. But again, there are no general rules. In other blood vessels, it can be different. For example, here, in human coronary arteries, you can see that bradykinin will not affect the cell membrane potential of vascular smooth muscle if the endothelium is not there, but if the endothelium is there, it causes a hyperpolarization. This is only one experiment, but it demonstrated that besides causing the release of nitric oxide, bradykinin also in certain arteries, not in all, can cause the release of VDHF and cause hyperpolarization and relaxation. And I don't show you data, but it also can cause a release of prostacyclin. Again, if you give a stimulus to an endothelial cell, it is very likely that that stimulus will act, we will see that later, through an increase in calcium concentration. And then all the activities of the cells are activated, most of them. See? And in this instance, the three pathways are activated. Now, in certain arteries, this pathway is not well developed, then it doesn't count. You know? In certain arteries, only NO is important, then it is only NO. In most blood vessels, the three can contribute to endothelium-dependent relaxations due to activation of bradykinin receptors. And Professor Dong Man Wu would tell you that this is a BK2, a 2 type, the second type of bradykinin receptors. It's not shown on this slide. Why do I insist so much on the importance of bradykinin? Well, because bradykinin is formed in our body. And bradykinin is also a substrate for a very important enzyme in our body. An enzyme that I've already mentioned, converting enzyme, that is located on the surface of the endothelial cells. And if you remember from the lectures on homeostasis, converting enzyme is the enzyme that transforms the inactive angiotensin 1 into the active angiotensin 2. You remember that? You remember that we discussed that? Yes, remember? You better. You have to remember those things. You know? Otherwise I get nervous. Good. But the same enzyme breaks down the active bradykinin into an inactive peptide. And again, I always say that, I may have said it already to this group, actually this enzyme was first discovered in Brazil because of this. Because it was a kinase, you know, it was breaking down kinase. Okay. Why is that important? Well, it is important because blockers of this enzyme are among the most prescribed drugs for the treatment of high blood pressure, of hypertension. I can guarantee you that if you know 5 people that have high blood pressure, 2 or 3 of those people still have or have been on an ACE inhibitor. And it's angiotensin converting enzyme, so it is abbreviated ACE inhibitor. Okay? Do you know such a person? Have you ever seen a person who takes every day an ACE inhibitor? You all have seen one. Me. I take one every morning. Okay? And that's one of the reasons why I became interested in the pharmacology of ACE inhibitors and of bradykinin, because, when the doctor tells you to swallow a pill every day, you try to understand why you have to do that. You see? So, good. And so, when we notice the endothelium-dependent relaxation of bradykinin, we wonder what would happen if we were blocking this enzyme. And in this experiment here, I show you in coronary arteries relaxations to increasing concentrations of bradykinin under controlled conditions and after incubation with the ACE inhibitor that I take every morning. Okay. And as you can see, there is a very significant shift to the left of the concentration response curve to bradykinin. In the presence of the ACE inhibitor, the blood vessel is much more sensitive to the endothelium-dependent relaxation caused by bradykinin.